things I'm really confused about. Maybe you can help me with that. Thank you very much. But that was too long for the slide. But that's basically the program for today. Okay, wait, Zoom is informing me of something I understand. Okay, cool. I wasn't exactly sure who my audience will be, so there will be a bit of general introduction in the beginning about something many of you may be familiar with. So the
Or is it not continuing? You've had that before.
Okay, I'm in control. I'm partially in control. Okay, no, I can make this work. Okay. In case you're not familiar at all with the alpha rhythm, I wanted to show you an example that maybe illustrates why I've been so fascinated with the signal, basically since I saw my first EEG recording many years ago. Of course, those of you who are working with EEG know exactly what I will be talking about. So this is a subject in an EEG experiment, and we will see the EEG signal recorded from her. And you can appreciate that there are moments when you can see easily this wave-like signal. But here, for example, that looks almost like a sinusoid wave, especially when she closes her eyes. And that's, of course, the famous alpha rhythm. And you can see that it's really a strong signal. There's no signal processing required. It's easily observable with the naked eye. And that makes it a relevant neurophysiological fact that we would like to explain. Certainly, if the brain goes through all the trouble of creating this very strong signal, surely that must be very important. And I want to understand better what it is important for, or more specifically, the effect of that signal, the correspondence of that signal with visual perception and visual information processing. Yeah, just saw that. So if you're busy and you need to leave early, no worries. I will summarize briefly what I am going to talk about. So I will show a series of experiments, one of which is about the effect of ongoing spontaneous alpha oscillations and how that changes what the world looks like to you. Then I will move on to experiments which I think are a bit more similar to what people in this institute are working on, which is changes in the alpha rhythm when people receive instructions to selectively process something. And I will show results from an experiment where we use that's exogenous attention, where we use exogenous cueing to induce alpha lateralization. And I will show that this is a rare situation where the focus of attention is anti-correlated to the lateralization of the alpha rhythm, for reasons I'm not entirely sure I've understood that. And last, I will show results from a short-term memory experiment that was originally designed to investigate the data format of spatial short-term memory, in which it also turns out that alpha lateralization is maybe not as involved in short-term memory maintenance as I previously thought. And then we will try to make sense of that. Okay, so a brief summary of what what I think I understand about the alpha rhythm. So, of course, it's been with us for a long time. It's been the first phenomenon, the neurophysiological phenomenon that was ever observed in EEG. And basically, from the very beginning, people had an intuitive understanding of the cognitive function reflected in the signal. And it's nicely captured in this quote from Adrian Matthews from the 1930s. The alpha rhythm shows what happens in an area of cortex which has nothing to do. This idea is often referred to as idling. You're not doing anything in particular, alpha is strong. In more recent years, we have learned that this idling, when the brain is not doing very much, this is related to reduced neuronal excitability. For example, as shown in this study, demonstrating that when alpha power is very strong, there is a drop in firing rates, spikes in this is in the somatosensory cortex. So strong power, reduced firing rates, reduced excitability. We also see that in humans. This is a monkey study. We see something similar in humans, for example, in fMRI studies, where strong power is associated with reduced bold activity. That's why it's blue in occipital cortex. The red spot is the thalamus, presumably because the thalamus is part of the pacemaker that drives this rhythm. And finally, many studies have shown that this suppression of neuronal excitability somehow has consequences for perception. This is one of the first studies showing that. This is a power spectrum. And if you don't know what that means, never mind. What this study showed was that when the faint stimulus that was difficult to see, when it was presented during moments when the alpha rhythm was strong, so there was physiological suppression, so to speak, then people reported fewer of these stimuli. They were more inclined to respond, no, I haven't seen this one. So now a lot of my research has been concerned with this kind of finding and trying to understand what that actually means and what is the specific effect of ongoing alpha and physiological suppression on perceptual decision-making. That's not a trivial question, because this finding that strong power means that people are more inclined to say, I really haven't seen this one. This could be due to a number of mechanisms. For example, it could be that when pre-stimulus, when ongoing alpha power is strong, there's more physiological suppression or less excitability, then maybe that reduces visual sensitivity, which for me, sensitivity in the signal detection sense means it impairs your ability to discriminate signal from noise. And that's why you miss many of these stimuli. That's possible. It could also be that the alpha rhythm somehow has an effect on how people make the decision what to report. Maybe similar to when you're a subject in my experiment. I tell you, look, you get €5 every time. Sorry, you have to pay me €5 every time you make a false report. You'd probably be more careful. This wouldn't change your sensitivity, how well you can discriminate signal from noise, but you would be more careful to say that, oh yeah, there probably was something. That's a change in decision strategy. Or alternatively, the state of the alpha rhythm and physiological excitability could have an effect on a subjective appearance, what the world looks like to you. And that's what the first experiment was about. And this was based on our previous finding that the primary effect really of the strength of the alpha rhythm at the moment the stimulus is presented is an effect on criterion in the signal detection sense. People respond more conservatively, more cautiously when the alpha rhythm is strong. And that's why they miss more of these stimuli. This is an illustration of a signal detection model that we developed to explain this effect that tries to give people an intuitive understanding of what this could mean. So suppose you are a subject in a detection experiment, and let's say we show this not the typical light dots or Gauss ratings, but actual images like these. And your task is to say, at this location, is there an object or not? So over here, for example, this would be a situation according to this model, what the image would look like to you if your alpha power was momentarily very strong. So there's a lot of suppression or not so much excitability. So in this model, this would result in a suppression of the signal. That's why I can't see very much. And a concurrent suppression of internal visual noise. That's why this image is very dark, but there's also no noise embedded in the signal. And now, if you have to report, what's going on here, is there an object? Yes or no? Then, well, no, you probably don't see an object. However, over here, you also see nothing. There actually is nothing. So you would not make a false alarm. So that's good. Not many hits, but also not many false alarms. Now, this is the same image. In a moment when your alpha rhythm is small, so there's more excitability, and according to this model, more excitability results in the amplification of the signal. That's why there's more stuff you can see here, but an equal amplification of internal noise. That's why this image is kind of blurred. And now maybe what you can intuitively appreciate is that now if you have to detect, is there something here? Maybe you have the impression that, maybe something is going on here. So you might say, oh, yes, I detect. And that would be a hit. This is actually a coffee cup. But you also see, maybe something going on here. So you might say, I detect something here as well, and that would be a false alarm because that's really just part of the noise. So bottom line is, according to our theory of how the alpha rhythm is related to perceptual decision-making, according to this theory, alpha power really has no effect on the signal-to-noise ratio, only on the amplification or inhibition of both signal and noise. So that was our understanding a couple of years ago. Now this interpretation makes a strong assumption, which is that, well, as illustrated in this figure, the strength of the alpha rhythm should have an effect of what the world looks like. Right? This image looks very different from that image. Now, if we did that experiment, like we already stay here in this room and we record your EEG, and then over time, probably your alpha rhythm will be getting stronger, or maybe it's already quite strong right now. And you pay attention to what this room looks like to you. I don't know. Maybe I'm relative. Well, at least I have never noticed that just sitting in a room there is a fluctuation in the intensity of what the room looks like, or in the intensity and signal and noise. And of course, we don't notice that because, well, first, probably this effect is relatively small. And also in a room like this, there are also changes in the ambient light. We move around, etc. There's too much factors to control for. So this needs to be investigated psychophysically. And that's what this first study was about. So here we wanted to test if the alpha rhythm has an effect on visual appearance. And more specifically, the experiment was designed to test the effect of alpha lateralization, the difference in power between the two hemispheres. And we adapted a paradigm that has been used a lot in psychophysical attention research, originally developed by Marisa Carrasco and colleagues, to study the effect of attention on visual appearance, on what the world looks like. Then our adaptation looks like this. So trial starts, and I don't have a point. The trial starts, people are fixating in the center. And there are these two potentially relevant stimulus locations. And the experiment comes into, there are two kinds of tasks that people do on different trials. In the contrast discrimination task, they fixate in the center and we present Gabor patches, left and right. And the task is to report which of these stimuli have the strongest contrast. And across different trials, we manipulated the actual physical contrast difference between the stimuli. On a separate set of trials, the stimulus was more or less the same, but we also presented a directional cue which instructed subjects, well, simply to report the orientation of the grating at that location. We use this as a kind of localizer because this manipulation is expected to induce strong alpha lateralization, which basically is kind of a correlate of the shift of your spotlight of attention to the cued location. So in that attention task, when we tell subjects, hey, pay attention over there, we find this strong effect in the alpha band. So the yellow blob here, that's lateralization, the difference between the ipsilateral and the contralateral hemisphere. And what this basically means is that if your target location is, say, on the left, and the right, that's not relevant, that's a distractor, don't pay attention there. Then this shows, and many other studies have shown before, that alpha lateralizes so that power increases opposite to the irrelevant stimulus. And it decreases in the hemisphere processing the relevant stimulus. That effect is very well known and very robust. And so we used this finding, this pattern of lateralization, these frequencies, these channels, as a kind of localizer to now quantify the spontaneous lateralization that was not instructed by a cue in the discrimination task. Sorry, one more thing about the timing. So here 0 means the onset of the Gabors, and the directional cue is somewhere over there. So in the discrimination task, we find significant lateralization just before stimulus onset when we compare trials where subject reported the left stimulus looks stronger versus the right stimulus looked stronger. And there was no cue involved. No direction was more relevant than the other. So this is a spontaneous effect. And so just before stimulus onset, we see this pattern of lateralization, which means, in other words, that suppose we show you these two stimuli and they are exactly the same, physically the same. And it just so happens that spontaneously, like by chance, your alpha rhythm is slightly lateralized. For example, you might have slightly stronger alpha power in the left hemisphere compared to the right hemisphere. This will have the effect that the stimulus that is processed in the hemisphere with more alpha power is suppressed, is subjectively suppressed. Or, who knows, the opposite. The stimulus processed in the hemisphere with lower alpha power is amplified. Or both. In other words, or in simple words, the distribution of alpha, the alpha lateralization, has an effect on your subjective contrast appearance of what the world looks like. Yes.
Can I ask something about this? Like, is it kind of a momentary effect, or is it a sustained effect, like when none of the items are cubed? Because, like, also from like Leibniz-Institut clinical potential literature, we know that if like there are possible changes in the environment at the same time, very equally likely. So even within seconds, like around again like 6 to 10 hertz probably, you sample from each side, and to follow the changes in the whole environment.
Right. So rhythmic sampling of attention would predict that, since you don't know, or since both locations are equally relevant, and you can sample only one at a time, you will be sampling back and forth. This may be going on this is a power effect, but it's not a phase effect. And there is let me look at the timeline again. Well, there is a stimulus that, you could speculate could evoke a reset in this rhythm, which is the onset of the central fixation course. But in the contrast discrimination terms, this stimulus would be all black. It would not systematically cause a reset over here and not over there. So I think it would not lead to this power effect that we see. We have not looked at phase effects. So I can only speculate.
Yeah, but for example, in terms of this specific study, we can expect, or we can say that even we have like two stimulus, none of them are corrected, but we still kind of select one of them and process it more. Right? Does it necessarily mean that because I have lateralization over one of us?
Whether one of them is selected, I suppose in the end, yes, because people have to make a decision about which one seems to have stronger contrast, and then report that. And I guess you could say that one is probably selected for report. Not sure that's an answer to your question. It is, in any case, the effect is short-lived. This happens only in the moment just before stimulus. It is not affected by post-stimulus activity. We made sure of that, but it's very short-lived.
Can I ask a small clarification question?
Okay. So this first experiment was about what I refer to as spontaneous alpha power, or let's say pre-stimulus power, in a paradigm where people are not specifically instructed to select any particular information. Now, this finding that the alpha rhythm is associated with a suppression of excitability and a suppression of visual performance has inspired a number of, and the finding that it can be so easily lateralized with cues has inspired a number of hypotheses about the cognitive function about this rhythm. So in this old formulation by Adrian and Matthews, the alpha rhythm shows what happens in an area of cortex, which has nothing to do. This sounds like a very passive phenomenon. That is something that happens to the brain when the brain is doing nothing. Why the brain does that, well, we don't know, but this basically states the facts and not more. In more recent years, there have been a number of proposals saying that, well, we should better think of the alpha rhythm as a tool. Those are my words, right? As a tool, which the brain uses to selectively suppress information that would be, task irrelevant or potentially distracting. For example, in this paper by Rees and Tudor and colleagues, saying that the task irrelevant space might be actively suppressed. The active, that's the important term here. Or in the gating by inhibition hypothesis by Jensen and Mazaheri, we say that this selective inhibition by alpha is used actively, purposefully, to route relevant information and to keep irrelevant information out. Sometimes I wonder whether these ideas are really different ideas compared to Adrian and Matthews, or using only different words, but we will come to that. So the idea is again illustrated here. So lateralization experiments leverage the fact that the brain has two hemispheres, the left and the right, and information on the left side of the nose are processed in the opposite, the contralateral hemisphere. And so, for example, if your target, the relevant information is on the left, it's processed in the right hemisphere of the brain, and the distractor on the right is processed over here. And now what would be really useful would be a mechanism that allows you to selectively suppress the distracting information. So to block this information from being processed. And the idea is that, well, that's exactly what alpha oscillations do for you. When you find that alpha power increases in the hemisphere processing that irrelevant stimulus. And effects that are, well, I would say that are consistent with this idea have been observed in a large number of different domains and paradigms. Of course, they are found with endogenous spatial cueing. When you tell subjects that look over here, now this will be very important. But also using exogenous cueing with cues that attract automatic attention in visual search studies, short-term memory studies, and many, many other studies. So it is certainly a very robust phenomenon. So in recent years, I would say that there has been an accumulation of findings that may indicate that maybe the story is not quite so simple. Alpha is this filter that filters out distracting stuff. For example, in many studies, it turns out that the strength of the alpha rhythm or the strength of lateralization is not really correlated with perceptual performance. With behavioral performance. Sometimes it is, no doubt, but often it's often not always, But sometimes it turns out that this lateralization does not really track where the distracting stuff is happening, which is weird if it's supposed to be a filter to filter distracting stuff. Rather, it's directed at the relevant stuff. That's not always the case. There are brilliant studies, for example, from people in this very room, where the effects are more consistent. Furthermore, sometimes the timing and topography of alpha lateralization does not really match the timing and topography of other indicators of attention, indicators that are also considered neural correlates of selective attentional processing, for example, steady-state visual evoked potentials. Finally, in recent years, it has turned out that the direction of alpha lateralization is surprisingly robustly correlated with oculomotor behavior. Now, of course, typically in those studies, subjects are very clearly instructed to not move their eyes and stay fixating on the central fixation cross. But of course, people do not always do that precisely. And in those studies that actually measure miniature eye movement, like microsaccades or small gaze shifts, it turns out that alpha lateralization is strongly correlated with these miniature eye movements. This is the backdrop for the first study about alpha lateralization I want to show. It's not much time left. In which we used exogenous cueing. And the reason for that was that most studies in this field of research that have used cues that instructed subjects to selectively process part of the screen have used cues that were expected to result in attention shifting to the cued location. Now here we wanted to use a paradigm where paradoxically the cued location is not where attention is, because attention is relocating in the opposite direction. And that's exactly what you typically find with exogenous cueing in the inhibition of return paradigm. So the paradigm and the task is very simple. People fixate in the center, and the trial starts with the presentation of the cue, and the cue is totally uninformative and irrelevant. People can totally disregard it. It really doesn't matter, but it's a strong stimulus, and this strong stimulus, it automatically attracts attention. Then there is a brief interval when the thing happens, and then we present a target. And the task is simply to, when the target appears, press the button as fast as possible. No discrimination. We don't want to know what the target was. Just press the button as fast as possible when the target appears. And the target can appear at the cued location or at the non-cued location. So behaviorally, this is what happens. I show you the response times, strictly speaking, the difference in response times between targets at the cued versus the non-cued location. Here's a subtract numbers in your head, never mind. So what you see here is how the response time develops over the temporal interval between the cue and the target. And what we find, and it's really just a replication of a well-known effect, is that when the target immediately follows the cue, response times are slightly faster at the cued location. It's early facilitation. But then as this interval grows longer, you see that the response time effect flips. And we find a rather sustained inhibition of return effect, which means that response times are actually faster when the target appears at the non-Q location. And this is usually interpreted as showing that when the Q appears here, attention briefly moves over there and then reallocates to the opposite field. And it stays there for a sustained time. Now, if this is the pattern, the behavioral pattern, the time course of attention, so to speak, where attention moves from here over there, and if alpha lateralization is a correlate of attentional orienting, then you would also expect that alpha gets lateralized towards the Q and then follows the behavioral effect. So let's see. So this is alpha lateralization shown separately for the different conditions depending on the temporal interval between Q and the target. And the most important panel is actually down here. That's a condition with just the Q and never any target. So you can appreciate that in all these conditions, there's this nice yellow blob. And the yellow blob means that at these early latencies, alpha is lateralized towards the Q. Okay, that makes sense, right? Sure, the Q attracts attention. We find behavioral facilitation at short intervals, so that's cool. And now we are looking for the relateralization following the reorienting of attention. And it doesn't happen. Don't find any. For example, in this condition where actually no target is ever presented, we quantify the strength of alpha lateralization at the different latencies where on other trials targets can appear. And this is plotted in this figure on there. Okay, so the black line is the response time effect, and you see that it drops very nicely. That's the behavioral inhibition of return effects, reorienting of attention to the non-Q location. And the purple line, that's alpha lateralization. And you see that it drops to 0, and then even while the inhibition of return is in full swing, it never reallocates along with the spotlight of attention. What happens as well, I have to speed up a bit. What happens as well is that while people do that, while we record EEG, people are also making systematic miniature eye movements, microsaccades. And the general direction of microsaccades is generally away from the Q, but they are correlated with the direction of alpha lateralization, meaning that the stronger my alpha is, directed in the Q location, the more my microsaccades are directed in the same direction. So that gives this strong positive correlation. So this supports other studies that maybe indicate that alpha is maybe more closely associated with oculomotor behavior than selective processing or orientation of attention. Okay. When did we start?
I think you can go like 10 more minutes or
Oh,
something.
10 is plenty. Okay, cool. All right, so third experiment was an experiment about lateralization in short-term memory. That's because many studies have shown that if I show you stuff and then tell you, actually, you need to memorize only this here, this also leads to alpha lateralization towards that relevant information. The primary purpose of this experiment was to explore the spatial coordinate system that is applied in the visual system. Basically, what does left and right mean? Left and right relative to what? Relative to the location on the retina, or relative to the location of the stimuli in the world on the screen. So to test that, I mean, that makes a difference if, oops, that makes a come on. That makes a difference if while you are memorizing something, Spider-Man over here, you are moving your eyes. Now that location where Spider-Man had previously appeared is now in the opposite hemisphere. The question is, what happens to your memory representation? Does it also switch over to the other hemisphere, or does it stay where it was? So that was the original purpose of this experiment. Maybe I will skip over some of the results we found. So this was our paradigm, pip fixated in the center, and we presented this bilateral array of colors, and people were instructed to memorize those colors, but only on one side of the screen, as indicated by the key. Then they memorized this information for a 1 second interval. And so far, this is a very typical visual short-term memory study. Now we show them another cue, instruct them to make an eye movement. The saccade to either the right or the left fixation block. And they had 1 second to execute the saccade, and then this was followed by the final post-saccade interval, and that's the interval we're actually most interested in. Finally, when they were probed, finally, they had to do a change detection task and report that this color is not the same as this color. That's the task. So one signal we looked at was the event-related potential. That's the contralateral delay activity. That's a signal that is very specific to short-term memory tasks. And the consensus seems to be that this is really a reflection of the actual maintenance process. Okay, there's a lot to unpack here, and in the interest of time, I will briefly summarize it, and you just have to believe me. So here, we see the activity during the maintenance interval before the saccade. And you see there's a strong difference between blue and red. Those are the conditions where you memorize the stuff on the left versus the right. And that makes a strong difference, and that's a lateralization effect. And that is the way we construct this. That's really the equivalent to the CDA. That's a signal lots of people have demonstrated in numerous studies. And now the question is, what happens to the signal when now subjects make an eye movement to the left or the right, so that the original location does or does not switch to the other hemisphere? And basically the finding is that you still see the difference between red and blue, And it looks more or less the same after a left or a right saccade. So for the CDA, the lateralization doesn't change no matter how you move your eyes. And that is indicative of a retinal topic spatial coordinate format. Okay, now we wanted to know what happens in the alpha band. Now, before the saccade, again, you can see that the difference between red and blue. In other words, in the way we set up this experiment, this is conventional alpha lateralization. That's what you guys are always showing. That's what everyone finds, maybe shown in a slightly different way. And again, the question is, what happens after the saccade? And it turns out after the saccade, there's no difference between red and blue, both after left saccades and right saccades. In other words, the signal loses its memory specificity. It's no more specific to what is being memorized. Instead, you see that the direction of the signal is determined by the direction of the saccade. This is, simply speaking, what these data show is that if this is the screen, and I make a saccade to the left, alpha always lateralizes back to the screen center. Same when I make a saccade to the right, alpha lateralizes back to the screen center. That's what's indicated by this lateralization pattern. We replicated this over 2 experiments. We also we correlated lateralization with individual memory capacity, tested in a separate set of trials. And while the CDA is strongly correlated with capacity, alpha is not. What it is correlated with is it's correlated with gaze drifts, meaning that it turns out that when we instruct subjects, say, to make a saccade to the left, if they make a saccade to the left, they land on the fixation target. Often there is a slight overshoot. And then the gaze starts to very slowly, very slightly drift back towards the screen center. This is a tiny, in terms of visual degree, this is a tiny effect. We're talking about maybe 1/10 of degree of visual angle, but it is very consistent. And the stronger this drift, the stronger alpha lateralization is back towards the screen center. So the conclusion seems to be that at least after the saccade, alpha lateralization isn't really involved in the maintenance of short-term memory. So to summarize, what I showed you reflects, well, a few things I kind of understand and a few other things that I'm not sure I do. So what many previous studies and some of our own seem to indicate is that strong alpha power indicates suppressed physiological excitability and a suppression of sensory appearance. When the alpha rhythm is strong, stuff looks darker or less contrasted to you. That's spontaneous alpha power. When we cue subjects to selectively process part of the word, then very often the direction of lateralization matches the focus of attention. But sometimes it doesn't. For example, in the inhibition of return paradigm, where paradoxically attention reorients away from the cue. And sometimes alpha lateralization seems to correlate very nicely with processes that also happen to be related to cues. I mean, they are probably part of our machinery of attention. But I think we can wonder if this correlation with oculomotor behavior if that is really captured by these interpretations of what the alpha rhythm stands for, gating by inhibition or active inhibition. So what this may indicate is that sometimes the correlation between cueing and the direction of attention might be more coincidental, rather than showing that alpha is a causal mechanism for stimulus selection, selective attention, memorization, etc. I'm not saying that there's nothing to it. So I want to make that very clear, that there are certainly studies showing that alpha is actually actively relevant for the selective processing of cued information, or the selective inhibition of irrelevant information. And many of these studies have actually been provided by your group. So when selective processing happens, probably alpha follows. What I am saying is that we should be careful doing it the other way around, using the observation that in an experiment alpha was lateralized somewhere, and doing a reverse inference based on that to infer that, well, this must probably show that there was selective processing going on, there was distractor inhibition going on, and alpha is the causal mechanism for that. I think I would be a bit more careful with this kind of interpretation. And with that, I thank my co-authors and I thank you for your attention.
Yeah, thank you so much, Niko. Yeah, I for sure have some questions, but I'm first gonna let the people here just to have said that if on Zoom anyone has a question, please use the raise hand feature and I'll try to coordinate and tell you when you can. So yeah, I know.
Thank you very much for the very nice talk and data. So my first question actually refers to the last part of your talk, but I would also have one for this IOR thing, but maybe at a later point. Now, what you've shown there and what I actually totally I really follow this reasoning and everything, that alpha lateralization is not so much about the selective storage of information in working memory. So you said that, okay, CBA, we know that it's this very typical correlate of selective storage, active storage of information in working memory when it's lateralized. And while I actively process this information and keep it activated in the system, you see the CBA effect, which also reflects